Namaste. Good day to all of you. This is Dr. Uma Kidvetal, Faculty of Agriculture and Veterinary Sciences, Jayoti Vidyapeeth Women's University, Jaipur. So I'm rejoining you with a session on agricultural microbiology lab this semester. And this session will deal with sterilization of nutrients. Sterilization in a microbiological laboratory. So in the previous session, we have learned about the various instruments that will be used in a microbiology laboratory. And also we have seen the parts and the principle of operation of a microscope. So now in this session, we will be looking into sterilization, that is removal of all contaminating microorganisms from anything that is to be used in the lab, be it plasticware or glassware or chemicals or medium components, medium as such, water. All of these have to be sterile. It cannot have contaminating microorganisms because we need to grow specific microorganisms in order to study their properties. So then if there are contaminating microorganisms which can grow at a much faster rate than what we intend to grow, these microorganisms will grow and we will not be able to get proper growth of the required microorganisms. So contamination in any way is not acceptable for an experiment. And so there are methods of sterilization that will be used in the microbiology laboratory. So I will first go into some of the important terminology that is used in the microbiology laboratory with regards to sterilization. So sterilization is the complete destruction or removal of all living microorganisms or their spores by any physical, chemical, or mechanical means, and it is called sterilization. So it can be accomplished by many means, like it can be using heat, and in that wet heat, dry heat, filtration, use of gases, irradiation. There are many modes of sterilization that is used in a lab. So the first and foremost will be sterilization by heat. So heat sterilization can be accomplished by several ways. So the first one is a red heat incineration. So this is used for inoculating loops and such. So inoculating loop contains a metal wire. So what we will do is it will be heated until the wire glows red hot. And then it will be taken out of the flame. So we'll be using a Bunsen burner for this. And once that is done, it will be taken out of the flame and allowed to cool. Then again dipped in alcohol. Please do not dip an incineration loop in the alcohol while it is red hot. The alcohol has got a like flame point, a very low flame point, and it will start burning. So please do not do that. But if you are like allowing the inoculation loop to cool and it becomes black again, and then you are dipping it in alcohol and sterilizing it, and then again heating it once more to remove all traces of alcohol, allowing it to cool again, and then uh, use it to take your microbial sample from the slant or the petri plate where you want to make a streak on the plate, then that is the proper procedure for use of a inoculation loop. So inoculation wire is something that is sterilized by using heat, red heat. Then flaming, I had told you the basic use of a Bunsen burner in a laminar airflow. Like whenever we are opening a conical flask or something, before opening, we will sterilize the mouth of the bottles or flasks or tubes or glass sides or test tubes such as by passing it through a flame without allowing it to become red hot. It will just be passed through flame for like a few seconds. It won't be allowed to become red hot. So that is the procedure for flaming. Then dry heat is used in a hot air oven. We will get into the details of it later. So in there also, it is used at like 160, 180 degrees. Heat will be used. Air will be passed through the hot air oven, and that is used to sterilize glassware such as glass petri dishes, glass pipettes, some metal instruments, and these are sterilized by using wet heat in a hot air oven. And then another method that is used, and that is the most commonly used method for sterilization in a microbiology lab, that is wet heat or moist heat. That is, this principle is used in an autoclave, in which we will be using heat or steam at 121 degrees Celsius and 15 psi pressure. So normal circumstances, it will be held for a 20-minute duration. If we are doing a decontamination, then it will be kept for much longer, like 30 to 40 minutes, or 2 cycles of decontamination for 20 minutes each for when we are decontaminating. Then there is the process of tyndallization. So it is a heating process in which some heat sensitive media or reagents are sterilized by steaming at like 100 degrees Celsius for say 30-35 minutes for 3 successive days to kill spores completely. So tyndallization is a method to neutralize the spores or decontaminate the media from spores. So like in the first occasion, the vegetative cells will be killed, and then whatever spores are germinating overnight, it will be killed on the next day, and then whatever is left will be killed, because they all of them will germinate by the third day and they will be killed. So that is the process of tyndallization. Then there is something called the ionizing radiation, such as X-rays or gamma rays. They are mostly used to sterilize plasticware, the disposable plasticware that is used in the microbiology laboratory. Like if you are using a disposable plastic petri plate, or you are using tissue culture plates, they are mostly in the disposable form only. So you are using that, or you are using sterile falcon tubes. So these will be pre-packed, and they would have been sterilized using ionizing radiation such as X-ray or gamma rays. Now, what about vitamins and such? Vitamin solutions, they get denatured at around 60-65 degrees Celsius. So they cannot be heated at all. So what do you do in order to sterilize such extremely heat sensitive material? That is done by the use of filters. So filters are available, like syringe filters are available, or otherwise filter papers are available that you can use in a filtration unit, and then use a peristaltic pump or another pumping mechanism in order to give a positive suction pressure and thereby filter the medium. So whatever particulate matter or microbes are present in that medium, that will get stuck onto the filter. So some sugars, blood serum, and media that are used for mammalian cell culture, they cannot be sterilized because they contain so many nutrients that are like so heat sensitive. And then so there are different types of filters, like the sintered glass filter or the seeds of asbestos filter, membrane filters that are like mostly the nitrocellulose filter, Chamberlain filter, Mander filter. These are all the kind of filters that will be used for the filtration process. So another one is the gaseous sterilization. So in that, ethylene oxide is used. So ethylene oxide is a highly toxic material for bacteria, fungi, and viruses, and also for most of the heat resistant spores. So if you don't have time enough to go for the tyndallization process, because tyndallization takes about 3 days to be completed, then it can be uh, like sterilized by using 10% ethylene oxide and 90% of rayon gas, and then it will be like freon gas, and that would be used for this purpose, and it is again very effective. So then there is also the cleaning of the surfaces. So mostly that will be the disinfection process. And for that, like when you're working with the laminar airflow chamber, you are working with cultures over there. So once you've done with the work, you will need to sterilize the workbench before and after your work. So for that, normally 70% ethanol is used. So the question will be, why 70% ethanol? Why not 100% ethanol? So that is because 70% ethanol has got more surface tension because of the uh, surface tension properties of the water. So that 70% ethanol can damage a bacterial cell membrane much faster than 100% ethanol, because 100% ethanol will dry off faster and it will not be retained on the surface of the microorganism long enough for it to have any bactericidal properties. So there are other antiseptic liquids that are used in the microbiology lab as well, such as the quaternary ammonium compounds like cetyl trimethyl ammonium bromide. That is also one of the most commonly used. It's a detergent as well. Iodine solution is used, hydrogen peroxide is used. Then another one that is used in the medical field, that is the chlorhexidine gluconate, that is used as an antiseptic in the clinical laboratories. So there are also some other compounds that are used for bactericidal properties in food materials and such. They're not following under the sterilization properties, but they are in the preservative or antiseptic properties. So manuka honey and such stuff will come in that. So now let us get into the common methods of sterilization. That will be dry heat sterilization and the wet heat sterilization. So coming into dry heat sterilization. So dry heat sterilization is normally done in a hot air oven. So hot air ovens can be operated from 50 to 300 degrees Celsius. At 50, 60 degrees, they are used for drying of your compounds and such methods only. But when you are going up to heat of like 300 degrees Celsius, then they will be used for sterilization purposes. So sterilization can happen from like about 1.5 to 2 hours at 160 degrees Celsius, or for a few minutes, like say 15 minutes at 190 or 200 degrees Celsius. And this is excluding the time that is required to preheat the chamber before the sterilization cycle. So please keep that in mind, that you have to have the chamber preheated. So there are the static gas air ovens and the flowing air ovens, in which like in the static there will be a heat source from underneath, and because the heat is coming from underneath, and you know that the hot air is having lesser density, it will move upwards. The cold air at the top will then move, be pushed downwards, and that will get heated, and that is how the circulation happens. But in the flowing air hot air ovens, there will be a fan also provided in order to make the movement of the hot air more uniform in the entire hot air oven system. So that is the principle of using a hot air oven for sterilization. Now, if you were to use moist heat instead, so I had already told you that that will be done in an autoclave. So there are two types of autoclaves. So those are the vertical autoclaves and the horizontal autoclaves. So the vertical autoclaves are what we use in the lab. They can either be the stout top autoclave. They are essentially a pressure cooker, and you will be keeping it until the first whistle, and then at that point, from there you will start counting for 15 minutes. It's a totally manual process. Or else there are the automated vertical autoclaves, in which it will automatic once we have done the setup, it will automatically do the preheating process until the pressure reaches 15 psi, PSI is pounds per square inch. It's an older method of measurement of pressure. So now it is kilogram per meter cube. And 15 PSI comes to slightly higher than 1 atmosphere pressure. And so at this higher than 1 atmosphere pressure, the boiling point of water will be 121 degrees Celsius, at which it will be boiling. And so it will be done. Now, the horizontal autoclaves are mostly used for industrial purposes. So they are much more convenient, and they will be made in huge volumes. Like if you have a horizontal autoclave of say 10,000 liters, like they will be of the industrial scale for use and sterilization of media for industrial use. And they will be so whatever the purpose for the use, moist heat or an autoclave uses moist heat in order to kill bacteria at 121 degrees Celsius and 15 psi. And depending on the requirement, the time might vary, but the normal required time is about 20 minutes. So that would be the principle of sterilization, and please go through it very carefully, because sterilization is an important process, and this is a preparatory process for all your experiments that will be done in the lab. Like if you want to grow any microorganism, you will need to prepare medium, and once you have prepared medium, you will need to sterilize it or autoclave it before it can be used for the experiment. So in the next session, we will be going into the types of medium that will be used, its preparation and its uses. So that'll be it for the session. Thank you. Have a good day.